All right, so you have here close to this axon bulb, right? Very close to your muscle at this place where is a neuromuscular junction. So you have these vesicles that are packed with acetylcholine, right? And we mentioned that those vesicles are made all the way up in the cell body, right? And what is the organelle that makes those vesicles? Golgi, yes. So everybody got the Golgi in your head? Not gonna forget it? Okay, Golgi. So this is the trans Golgi vesicle that is putting these little packets all the way down to the end of the axon, right? And do you remember how acetylcholine gets into the vesicle? It's made up of acetyl-CoA, right? That is a byproduct of glycolysis, right? So you have acetyl-CoA and you have choline, and that comes from the food. A lot of foods are rich in choline, right? So you get acetyl-CoA and choline. So you have an enzyme here in the cytoplasm, right? You have an enzyme in the cytoplasm that put these two molecules together, actually only the acetyl part of acetyl-CoA and the choline part of it, and you make acetylcholine here in the cytoplasm. And then the question was, how is it that you put it inside of the vesicles? How does the thing get inside the vesicle or past the membrane? First thing that should come into your mind is you should have a transporter, right? So you should have a channel. You should have a channel for this thing. Now, the problem is that you cannot just open this channel and allow acetylcholine to go in, because the concentration of acetylcholine is a lot higher inside this vesicle than it is outside in the cytoplasm. So if you just open this channel for the fusion to happen, what's going to happen? It will leave. Yes, that's a natural thing. If you are in high concentration in one tiny space and you have all this big space out there with not much of itself, you're going to naturally leave the vesicle. So passive diffusion is not an option here, right? You have to do some kind of active movement of this molecule, right? But it's not a direct active transport, because you're not going to use ATP right in this channel right here. Okay, so it's not direct usage of energy here. What's going to happen here is not a primary active transport, right? This is a secondary active transport. So you have to figure out where is the primary active transport happening. Okay, so there is this other protein at the membrane, right? Of course, there are many of them. There's not only one, right? There is this channel here that is called a proton pump. Okay, so since it is a pump, it's going to concentrate things inside this vesicle. It's going to pump protons to inside of the vesicle. Okay, so this proton pump here is going to get a proton that is out in the cytoplasm, use ATP hydrolysis as the energy source to pump this proton to inside of that vesicle. Now you have a proton gradient that is inside of the vesicle. Now this channel can open and function as an antiport. Antiport because a proton is going to come out and is going to exchange that proton coming out with an acetylcholine that is going to go in. How is it that the proton is going to go out? Is it active transport or is it diffusion? Active transport over here, ATP usage. What about there? That's diffusion. That's the only other option, right? So active transport happen over here. How? Use ATP energy to pump this proton to the inside of the vesicle. That is your active transport right there. This pumping action concentrated protons inside of the vesicle. Now the protons, if something opens and it can go through, right? These protons are going to go down concentration gradient. So that is a secondary active transport type of transport, right? But it just doesn't go out because this thing here is designed to be an anti-port channel. So as this proton goes out, one acetylcholine gets in. Okay, it's 1 for 1. One goes out, one goes in. Proton goes out by diffusion, pushing acetylcholine in by just pushing it. I don't know how. It's pushing it. Okay? So this is your anti-port system. This is how this thing gets there. Another thing that was important that I mentioned here, once you have your vesicles all packed over there, right? You have your vesicles all packed over here. Now, once you get a signal from this nerve that it's time to activate your muscle, you're going to fuse this vesicle with the membrane to release the acetylcholine that is inside, right? Now, notice that the fusion is going to happen at this side here of the membrane. It's not going to happen up here or up somewhere else. It's right at this side of the membrane. Then you should be asking, why? Why not somewhere else, right? There's membrane everywhere. Why does it happen over there, right? Well, basically, this side here of the membrane, it has some little structures that are proteins. I'm going to draw one here. Okay, so these structures are proteins, and at first they are blocked. So you have one here, and you have another type of protein right there, the vesicle. Okay, so at first this protein here is blocked, but once you get your action potential that travel all the way the axon, right? Remember, this axon is myelinated, right? It traveled through saltatory action potentials, right, all the way down the axon, and then it reached, see the sodium channels opening here, so that it reached here, this side of the membrane of your axon. When this action potential gets there, the depolarization, when it gets to be positive enough, it is going to cause this voltage-gated calcium channel to open. Okay, so this is really important. It's a calcium channel that is going to open. Okay, once this calcium channel opens, of course, calcium is going to get into the cytoplasm, right? And then this calcium is going to get here and is going to associate with this protein at the membrane. It's going to unblock it. Okay, so calcium gets there, unblock this protein. The name of this protein over there at the membrane is called T-snare. Okay, T is snare because it's a target snare. Okay, and the name of this protein that is in the vesicle here, what are you going to call it? V snare, because it's vesicle. Okay, so this guy here is a V snare. So now they are able to interact because they're not blocked anymore. And when they interact, they bring this vesicle with very close proximity to the membrane. Okay? Once the vesicle gets very close to the membrane, then the membrane is going to fuse. And the fusion is going to open a hole in this vesicle and is going to allow the acetylcholine to get into the synapse. Everybody got that? Okay, and this is really interesting because when you think about this type of fusion and receptor mediated type of fusion, this receptor here is inside of the cell, right? But we see the same type of mechanism when you look at other things, for example, viral infections, and more specifically with enveloped virus infections, right? You know, we have two types of viruses. You have enveloped virus that they take a piece of the membrane and they use it as a coat on themselves. Who took virology or know enough about viruses? You took virology? So you know these viral enveloped viruses, they basically camouflage themselves with a piece of the membrane of the host, right? Now, when they're going to infect other cells, they usually have these proteins that interact with the receptors of certain cells, and then the proximity of the receptor binding causes the proximity of the membrane of the envelope virus to the target cell, and that's how a virus can infect a cell. Okay, so by the same type of mechanism that you see vesicles being dumped in synapses. Okay, so it's not something that is exclusive for this type of work. Okay, other things use this type of exocytosis or endocytosis, fusion of membranes, basically. Okay. All right, so here, what caused the fusion of these vesicles? It was the inflow of calcium. Okay. So I had a couple of students who came to my office and said they really didn't understand about this action potential business, and they're quite really confused about it, which I understand. I feel your pain. Okay, so we're really going to make sure you understand that. Okay, so what is happening here? The action potential that came down the axon is going to cause a change in the polarization of the inside of this membrane here, which is also an action potential. It's going to become an action potential too. But at first, this is called an end plate potential, end plate potentiation, or end plate potential, okay? So let's look here at first what is happening with the action potential at the neuron, okay? So at the neuron, the first thing that happens is that you have here your cell body. Okay, so this is a pretty neuron here. I know it looks kind of like a hand, but it's really, okay? It's the nucleus. I'm not gonna draw much here. So here at the dendrites of the neuron, you're going to have little signals that are going to tell this membrane to depolarize. Okay, what is the resting potential of a neuron approximately? Minus 70 millivolts, right? So that's the potential inside of the membrane. So if this neuron, these dendrites get one little signal, then it might not be enough to cause a lot of change of that membrane resting potential. Okay, so this same dendrite here can get several signals like in sequence. Okay, so the name of those signals, those are called temporal summation of those signals. So that means you're at the membrane. So here's the dendrites. Okay, so here's a dendrite, and I go, dendrite, move. Dendrite doesn't care. Move. Doesn't care. Move. So this is temporal. Okay, it's in series. Okay. So now this is one type that you can accumulate these signals to really try to push the inside of the membrane to become less negative. Okay, but it still might not be enough to cause an action potential in that neuron. There is another way that you can get these signals together to really push this thing to work, and that is called spatial summation. So that means when this guy gets a signal, so something else sends another signal to this dendrite over there. Okay, so it's at the same time. So here's another dendrite, and the neuron is the table, neuron body. Okay, so here we go. Move, move, move. So now we have temporal, and you have summation of the spatial part. Okay, so we have temporal summation and have spatial, because two of them are getting the signal or some signal at the same time. Okay, so now we go move. Eventually, this thing is going to get so annoyed that it's going to send an action potential down the axon. Okay, it's going to change that inside of the membrane. It's going to become more positive. All right. So what happens at the action potential? What is it really? At first, you're going to get sodium channels to open, right? And those sodium channels, they are voltage gated sodium channels. Okay? Remember how the voltage gated sodium channel looks. It has a little ball stuck to a chain that's going to inhibit the channel as soon as it gets activated. It's very quick. Okay? So first you have your sodium channel, this is your voltage gated sodium channel that opens, and that causes a flood of sodium to the inside of the cell. So your voltage is going to go from more negative to more positive. Okay, so your voltage goes up here. At that time, when you have this change in voltage, at that time you're going to have your potassium channels to open. Now here, I know it's a bit counterintuitive because you're thinking the inside of the cell should be down, and then this thing is showing the thing going up. But what is happening here is really track track of the voltage inside of the membrane, okay? So first, sodium goes out because it's in more concentration inside of the cell, right? So it's more concentrated inside. If you open the channel, sodium will go out. As soon as it opens very quickly, you have that inactivation ball that gets the channel stuck and the channel will close. So at that time, you're gonna have your potassium voltage gated channels that are going to open. Now, where do you find more potassium? Inside of the cell or outside of the cell? Inside, why? Why do you have potassium inside of the cell more than you have outside of the cell? Yeah, which pump? Sodium potassium pump, right? Really, very important. Everybody got that? Sodium potassium pumps the sodium out, 3 of them, and 2 potassium in. 3 sodiums out, 2 potassium in. All right, make sure you absolutely know that. So how many sodiums go out? 3. Let's do the sodium potassium pump exercise. 3 sodiums out, 2 potassiums in. 3 sodiums out, 2 potassiums in. Everybody's tired today. All right, hands up. 3 sodiums out. 2 potassiums in. 3 sodiums out. 2 in. You're gonna get it right, right? Good. Okay, so now when you have potassium going out, is potassium going out? 3 sodium out, 2 potassium in. That's the pump, right? When you open the potassium channel, what's going to happen to potassium? It's going to go in. Oh, you see, that's why you're going through the exercise. Because we were talking about the sodium potassium pump. The job of the sodium potassium pump is to put 3 sodiums out, 2 potassiums in. That is going to keep your resting potential at negative 70, right? Forget about the sodium potassium pump. Look at the membrane. If you have a channel there and you open a potassium channel, what's going to happen to potassium? How many people think it's going to flow out of the cell? How many people think it's going to go into the cell? It's going to go out. Why? Because you already pumped it in with the sodium potassium pump. So you have a high concentration of it inside of the cell. If you open a hole in the cell, potassium is going to go out. Yeah. No. What's going to happen to sodium first? Where did the sodium potassium pump the sodium? Out. So where do we have more sodium? If you open a hole in the membrane, what's going to happen to the sodium? It's going to go in, right? So when the sodium goes in, what happened to the voltage inside of the cell? It gets more positive, right? It gets more positive. That is what is meant by this line going up in the graph. Sodium channels are open. Inside of the membrane gets more positive. Now remember this potassium channel guy, it was lazy. It was a bit, delayed response type of thing, right? It took a little time to open, right? But when it opened, the potassium will get out of the cell. Potassium is what charge? Positive. If something positive gets out of the cell, what's going to happen to the inside of the cell? Everybody sees that the inside is going to get negative if something positive is getting out, right? So look what happens. When potassium channels open, this voltage goes down. Does that make sense? Yes. So when the sodium channels open, the voltage goes up. right, inside of the cell because sodium is positive and it goes to the inside of the cell. When potassium channels open, the voltage goes down because potassium is positive and leaves the cell, right? Okay, and then it goes down a lot and then it stabilizes back to resting potential. Now, it goes down a lot because if the sodium potassium pump here is always working. Okay, sodium potassium pump is always working. So here, the sodium potassium pump at this point is really trying to compensate to keep this thing back at resting potential. Okay, and what happens is that it compensates to the point that it reaches lower than the resting potential. So resting potential, let's see, is like minus 70. When it goes down after this potassium channels open, it goes down to like minus 90. Okay? How is it that that happens? It happens that so much potassium leaves the cell that the membrane really goes down a lot. The membrane potential really goes down a lot. Okay? Now, what happens here is that after you have the sodium potassium pump, pumping 3 out, put sodium 2, potassium in. So what happens here is that you always have some little leakage channels of potassium that are open, but just a little bit of potassium gets out. Okay? And that stabilizes the membrane potential at negative 70. I hope that makes sense. If it doesn't, come to my office or drop us an email or something, or ask a question. Okay? You always have a little bit of potassium channel leakage. You don't have sodium channel leakage. You have potassium channel leakage. Okay? All right, so that action potential got all the way here to the end of the axon. Open these calcium channels. Calcium channels cause fusion of these vesicles to the membrane. Now these vesicles are going to dump the acetylcholine at the synapse, right? And what's going to happen at the synapse? You are going to get ligand-gated ion channels. Is mainly a sodium channel, but a little bit of potassium is able to go through that too, but it's mainly a sodium channel, okay? It's going to get this ligand-gated sodium channel. Is actually called cholinergic for acetylcholine, but this is called a nicotinic cholinergic sodium channel, basically. Okay, so 2 of those acetylcholine bind to that, open the channel. What's going to happen here? Let's go to the next. Here. So this is what's going to happen to the membrane at your end plate, at your neuromuscular junction. Okay. After this chemical release of stuff from the neuron bound to this chemically sensing channel, channel opens, sodium goes in, the inside of the membrane, the same story that happened with the neuron, but now it's the muscle. Okay, the inside of this membrane, which one is this? Is this membrane? What's the name of the membrane? Sarcoplasma, sarcolemma, something like that. Sarco something has to do with the muscle, right? So this membrane here, after you got a flush of sodium here, it became positive on the inside, okay? So this thing here is called end plate potential. It's a little bit different than your action potential, because the action potential had to do with the voltage-gated sodium channels, voltage-gated potassium channels. This guy here have ligand-gated sodium channel, right? So it caused this depolarization of the membrane, okay? Now, just past this center here close to the synapse, you're also going to have voltage-gated sodium channels. So this thing is going to become an action potential at the muscular side. Okay, so here is just showing the flow here of sodium, a little bit of potassium, but it's mainly this flow of sodium that depolarizes the membrane. Okay, so here comes your first question. Oh, please work, please work. All right, so let's see. It is working! Yes! Okay. Yeah, sorry, I got a bit excited. At the neuromuscular junction, the muscle contraction initiation event is what? Pay attention to the question. Here it says, at the neuromuscular junction. Where is the sarcoplasmic reticulum? Is not at the neuromuscular junction, is down the line. Down the line. Okay? So the first thing that happened, initiation event, so let's see this. Conduction of electrical tube. Oh, this is the same as that because the T tubule is part of the sarcoplasmic reticulum. So B is as wrong as A. Binding of acetylcholine to membrane receptor on the sarcolemma. Sarcolemma is your plasma membrane of the muscle, right? And we just talked a lot about acetylcholine. Sliding actin, hopefully nobody answered. Yes, nobody answered that one. Good. So your correct answer is C. So at the neuromuscular junction, you're going to have acetylcholine that is going to stimulate those ligand-gated channels. All right. So here, you know that you're not supposed to keep acetylcholine at that synapse for a long time. Otherwise, you're going to keep on stimulating the muscle, right? And that's not necessarily a very good thing. Okay, so you got to have those acetylcholine really being degraded very quickly. Now, a way to take neurotransmitters away from the synapse, so it was like a way or a manner to take neurotransmitters away from the synapse. The thing can be done in several ways. One way is to get the presynaptic axon to reuptake a neurotransmitter. Another way is to break down the neurotransmitter, and still a different way is to get the neurotransmitter to diffuse away from that thing, far away from it, okay? So at the neuromuscular junction, the two main mechanisms of getting rid of acetylcholine is, first is degradation, because of this very fast enzyme that is present there. The name of the enzyme is something that ends with A's and kills acetylcholine. So it makes sense to call the enzyme acetylcholinesterase. Okay, so that's your enzyme. It's very fast and is present at the synapse, neuromuscular synapse. Now, diffusion is also possible, but usually the enzyme is a lot faster than the diffusion at the synapse, the neuromuscular junction. Okay. Reuptake doesn't really happen much, or doesn't really happen at all at the membrane, at this synapse. So you shouldn't really worry about that. Okay. So main mechanism is breakdown of acetylcholine by cholinesterase, this enzyme that is right there at the membrane, postsynaptic membrane. Okay. Now, look at this. Action potentials will not stop until the acetylcholine is removed, okay? If you have acetylcholine in there, without breaking it down, you're gonna have a lot of muscle contraction, a lot of muscle action potential, okay? So that means your muscle is gonna be contracted all the time. It's not gonna relax, okay? So you have a constant contraction of that muscle. That is called tetanus. Okay. Okay, another question for you. What would be the first response of a muscle fiber, that's a muscle cell, right, treated with something that blocks your enzyme acetylcholinesterase? Gotta think about that. Now let's think about it. You're blocking the enzyme that cuts down acetylcholine, right? So the first choice here says there will be a continued muscle fiber contraction in the absence of additional stimulation. You don't have to dump any more acetylcholine at the memory. And then I had a lot of people who answer here. The muscle fiber would continuously contract for a prolonged period of time. Oh, but see, this is one of those kind of tricky questions that I'll never put in a test, right? Because this one here, you have to think, maybe this thing is continuously contracting because you're sending down a lot of stimuli, okay? So the most correct answer here would be you continue to have contraction, but without additional stimulation, because the neurotransmitter did not get broken down, okay? Yeah, this level of question is a bit more challenging than what I like to use. Okay, so here we talk about the depolarization, right, of the membrane. So that's going to be your action potential. Now, remember, when you see this sodium channel with a ball and a chain, that means this guy has this inhibitor, and this guy is a voltage-gated sodium channel. All right. So remember, that's the guy that opens first, and then almost instantly it gets blocked, and then later you have potassium that opens, and then potassium goes out, and then later on potassium channel gets blocked too. Okay. So for this thing to happen, for you to get the sodium channel to open, you have to reach a certain voltage inside of the cell, inside of the membrane. Okay. So that certain voltage is called the threshold. Okay, the threshold voltage that is going to trigger that sodium channel to open. Okay. Once the sodium channel opens, you cannot stop the action potential from going down the cell. It's an all or none type of situation. If you get one channel that's going to open, they all are going to open. Okay, because you reach that threshold, and then your membrane potential is going to be even more positive, and that's just going to cause all the channels to open. Okay, so this action potential is not stoppable. So now we're going to have here the next step, that is repolarization, right? So this is when you have your sodium channel that is blocked. Your potassium channel is still open, but it's going to close too, right? And then you have your sodium potassium pump working, and that is going to get your membrane down to resting membrane potential. So you cannot reactivate this muscle fiber until you complete this process of repolarization. So this is called refractory period. Okay. Remember that we went into more detail about absolute refractory period and relative refractory period. There is a little bit of difference in there, right? You have absolute refractory period when your sodium channel is blocked, is inactivated. That's your absolute refractory period, because you cannot open the channel. You're not going to get another action potential, okay? The relative refractory period is when this ball comes out of there, but the channel is closed. But you don't have your membrane potential at -7. You have it, for example, at -90, like way down. Okay? So if you draw here your action potential, this is time, and this is millivoltage, right? So here you have like summation, 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 and then whoo, what happened there? Sodium channel open, and then whoo, potassium channel open. And then you have this kind of thing that goes to -70. So this is -70, this is -90. So this overshoot here, the membrane gets hyperpolarized, right? This time here of hyperpolarization is a time of relative refractory period. Because after this membrane potential went down from -70, the sodium channel unplugged, but it remains closed. It's just going to open after it reaches a certain voltage, right? Positive, not negative. Now, or less negative, I would say. Actually, it's like -55 when it opens, but this is not drawn to absolute certainty. Okay, just to give you the main idea of what's going on. So when your sodium channel is unplugged but closed, and your membrane potential is lower than your normal membrane potential, that's called your relative refractory period. Okay? When it's blocked, that's your absolute refractory period. Gotta know what happens there. So here is what is happening at the muscle fiber. So the muscle fiber, actually the resting potential is even lower than at the nerve. So this is like minus 90, okay, it's even lower. So what happens here? When you get the signal from acetylcholine and then your influx of sodium, then you get your voltage gated channels that open, right? So your membrane gets depolarized, and then your sodium channels close, potassium channels open, just like it happened in the neuron, okay? And you're gonna get repolarization, and then when your potassium channels close, then it goes back to resting potential. Simple. The muscle is a little simpler than the nerve. Okay. Oh, another question. Told you, got tons of questions today. In a muscle fiber, the key intracellular event that stimulates a muscle contraction is known as, most people got it right, it is depolarization. Okay, so here we're gonna really look at what is happening inside of this muscle. Okay, that's the other part, this other concept that you absolutely have to know. You have to understand, memorize it and eat it and really, really internalize it. Okay. So remember here you have your muscle fiber, right? The fiber is the cell, right? So here is a blown up version of your muscle fiber. Remember your muscle fiber that is voluntary, is striated and has a lot of nuclei, right? And you're going to differentiate voluntary muscle fiber from heart muscle. That heart muscle is going to have one nucleus, right? So this one is a lot of nuclei and very, very long fiber. Okay. So here you're looking inside of the cell. So here you have the cell membrane, right? And then here inside of the cell, you're going to have this myofibril, right? So looking here at the myofibril, so you have a lot of them that are composing your cell, right? Looking at the myofibril, remember the smallest unit for muscle function is called the sarcomere, right? So now we're going to look inside of the sarcomere and see what's happening with those proteins that are in there. So we mentioned you have the thick filament that is made up of myosin protein. So you have a lot of this myosin protein. So basically each of these microfibrils can have about like 1,500 thick filaments and about 3,000 thin filaments. So you have a lot more thin filaments than thick filaments, right? So here, when you look at the thick filaments, remember myosin, and myosin has a head, it has a tail, it also has a neck, very important for the bending function of that myosin. And it has a lot of them. And then here the actin, you have filamentous actin, that is F-actin. So you have this double chain here of actin, it's kind of double helix formation, right? But you not only have actin, you're going to have troponin and tropomyosin. Troponin and tropomyosin associated with the actin. Let's see what these things are doing, okay? First, when you are going to contract your sarcomere here, you remember those, first the definition of the sarcomere is between the Z lines. Z lines. Z line is the thing that looks like a zigzag. So between the Z lines, you have a sarcomere. Protruding from the Z lines, you have your thin filaments, and then in the middle, you have your thick filaments. Okay? So this is what's going to happen here with your sarcomere. So this is the Z here, there. You're going to slide the thin filaments together, and you're going to have the thick filament that is just going to associate with the thin filament, right? So the thick filament is going to be sliding that way. The thin filament is going to be getting closer together. All right, so what's happening with the bands here? If you're going to look at the bands, so first remember between Z and Z, that's a sarcomere. So you have two sarcomeres. Right? So you have here your age zone, that this is a place that you have only here your thick filament, right? Your age zone is going to shrink. Then you have here your I band that is also going to shrink. The I band is the part that is thin, right? Remember I for thin, okay? So I band is going to shrink. Now the A band is not going to change size because that represents only the size of your thick filament, okay? A for fat filament, okay? The I for thin filament and A for fat filament, but it's really the thick filament, okay? So let's see what is happening here mechanistically speaking. So looking here at your thick filament is made up of a lot of these myosin proteins. So each unit of these myosin proteins is really made of two myosins here put together. Okay, so the tails are intertwined. It kind of looks like a DNA molecule, not DNA. Okay, just myosin tail. Okay, and then it has two heads. All right, it kind of looks like a conjoined tadpole kind of thing. All right, so it has two heads. And then in the heads, you're going to have two very, very important sites. One site is going to bind to actin. The other site is going to bind to ATP. So the thick filament is really what's going to use energy in your muscles. It's going to break down the ATP and make this thing move. Okay? So that's the thick filament job. And keep in mind that you have a lot of them that is in one thick filament. A lot of those are associated there. Okay, so you have the helix, that is the tail, and then you have these globular heads. You're going to have a third part, oh, it just went back, of that troponin. Let me get there. There we go. This third part here of the troponin is going to bind calcium. Very, very important to know that. So troponin is not a thread, it's a blob that has 3 balls in it. One of them is going to get stuck to actin. Another one is associated with that, is all 3 parts, right? Another one is getting stuck to tropomyosin. It's going to anchor it on the actin. But the third part of this blob is going to sense different concentrations of calcium. Because when the calcium concentration goes up, then this thing is going to bind calcium and it's going to change conformation. That's a very important thing to keep in mind. So here is your thin filament. Remember, actin can come in 2 forms. One is globular, and this is actin. We're not dealing with that now. We're dealing with the filamentous F-actins, right? 2 helixes here, two threads together. So filamentous. So here you have your filamentous tropomyosin. Look, it's also made of 2 things, right? Okay. Tropomyosin, and then you have your blobs of troponin. When they get together, you have here troponin that binds to tropomyosin and to actin, right? The anchors in there. Now, the job of tropomyosin is to block the binding site of actin. See all these black dots? Those are the binding to what? What's going to bind there? The head of myosin, right? So if the binding site is blocked, so the head of the myosin cannot bind. So here's the whole idea, right? A low calcium concentrations, you're going to have here blockage of your active sites, and myosin cannot make a cross bridge with actin. Okay, everything is quiet there. All right. So now let's say that you have an action potential that came down the neuron, right? The motor neuron. Now you have depolarization here of your muscle fiber. This depolarization, this action potential, is going to propagate. You see the sarcolemma right there, the membrane, right? Is going to propagate to the sarcolemma and is going to go inside these invaginations of the sarcolemma. That is not, they're not showing here, but it really goes inside of the fiber, okay? So what that's going to do is going to be in contact with your sarcoplasmic reticulum, the blue stuff over there. The sarcoplasmic reticulum is derived from your smooth endoplasmic reticulum, okay? And the job of that cytoplasmic reticulum is to store calcium. Okay, it's going to keep calcium in there. Why? Because when it gets a signal of depolarization, it's going to let the calcium out. Okay? And it is strategically positioned this way that goes transverse in relation to your sarcolemma here. Okay, it goes transverse to that because it has to activate the fibers. It has to activate those things that are stuck to your thin filaments. Okay, so once this thing gets the signal, it propagates down here to these T tubules. T tubules are just extensions of your plasma membrane, and they're called T tubules for transverse tubule. They transverse inside the cell. Propagates down the T tubule, activates here release of calcium from the sarcoplasmic reticulum. And this is the stuff here that is bold and red. Okay? So this is a better picture of what is happening here. Here comes your action potential, comes down here. See, here is a hole in the membrane. That's your T tubule right there. You see, it looks like a little tube too. It's a hole in the membrane. The hole in the membrane here is surrounded by sarcoplasmic reticulum. They're in close contact. So depolarization of this hole in the membrane is going to depolarize here the sarcoplasmic reticulum on both sides, right? That depolarization is going to allow calcium to be dumped in the cytoplasm. It's going to increase the cytoplasmic concentration of calcium. Right, everybody got that? I just hope I don't sound like that teacher in the Charlie Brown. I always have nightmares with that. You know what I'm talking about, right? Blah, 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 blah. And then everybody's like, oh, you know what I mean? Okay, is everybody awake? Yeah, wake up check. Yeah, no? Okay. So here, let's shock this membrane. What's happening at the end of the axon? Signal that comes from where? Think. Somebody, please. Where's the signal coming from? Your brain. Hopefully. Okay, so signal goes down the brain, goes down the axon, simulates here, action potential. Action potential is going to travel on the membrane, penetrates this fiber. Sounds painful. Stimulates endoplasmic reticulum that is really called sarcoplasmic reticulum to release calcium. Where is the calcium going to go? Is it going to bind to tropomyosin or to troponin? Troponin. Why? Because troponin doesn't have myosin on its name. No, that's not why. It's a conformation, right? Calcium is going to bind to troponin. Troponin is going to be the calcium sensor. Once it binds to troponin, right there, is going to change conformation, but because that thing is an anchor of tropomyosin, when troponin is here quiet, doesn't bind calcium, is relaxed, hold here to tropomyosin, blocking all the active site stuff. Okay. When it binds calcium, these things go, this little yellow thing, troponin goes. So when it goes, it pulls tropomyosin away from that binding site or your myosin. Everybody got that? All right. So calcium is going to do that thing. Okay. Now, myosin is happily gonna bind to actin. That's what it's meant to do, okay? So here, another picture here of showing the invagination of the T tubules and this beautiful cytoplasmic reticulum, okay? T tubule, remember that, cytoplasmic reticulum. Where the cytoplasmic reticulum comes from again? Smooth ER. Just in case you don't remember, it's a smooth ER. Look at it. Does it have little dots? No, so it's not rough. It's smooth. Okay. Another question. Told you I had tons of questions today. The sarcoplasmic reticulum is an elaborated. Oh, that's not fair. I just told you the answer. All right, let's go quickly. Smooth ER. Oh, another one. Hey, who knew? What is the significance of the muscle fiber triad relationship? I briefly mentioned that. Let's see if you got it. Your answer here is D. Heat tubules conduct electrical impulses that simulate the calcium release from the sarcoplasmic reticulum. All right. So here, in detail, this is very, very important to know too. You absolutely have to know this cycle. Okay, absolutely. That's why I'm spending time on it. A low calcium concentration, nothing moves. Simple. All right. So we have tropomyosin that blocks the active site here on actin. So just keep this picture in mind. And remember, you also have here your troponin that's holding on to tropomyosin at that position. Okay. Now you have action potential. You dump calcium in the cytoplasm, right? You dump calcium in there. That is going to bind to troponin and cause the motion, right? So that exposes your active site. Now this guy here, myosin head, it doesn't have ATP stuck to it. Okay, so it has ADP, that's it. So when it has ADP in there, so let's make sure we mention that it has ADP attached to here. Now it has high affinity to bind to that little active site that is meant for it to bind on actin. Okay, so that's what's going to cause myosin binds here to the actin. When it's bound, this is what is called cross bridge. When it's bound, that's a cross bridge. Okay? So bound is a cross bridge. Now, it's only going to be bound when it has ADP attached to it, not ATP, right? ADP. Okay, so now what's going to happen? Remember, calcium has to be there, so the active site, binding site is not blocked. Okay, so this is in the presence of calcium. Once it is bound, because of this binding, then myosin is going to change conformation. When it changes conformation, it kicks out the ADP. Okay? Once it kicks out the ADP, it has an empty space there that is in the right conformation to bind to ATP. Does that make sense? Yeah. It comes from the circulation, from the blood, from glycolysis, from oxidative phosphorylation that we're going to see next time. Okay? Mitochondria, the muscle is full of mitochondria. Okay? And especially fatty acids and yeah, all the good stuff that gives us energy. Okay? So the ATP comes from there. Okay? Yeah. When the ADP is gone here, this is when the head does the thinking and the pulling part. Okay? So when the ADP is there, it binds, ADP gets out, and this thing cranks. And it's bound to actin, so it's going to move the actin. Okay? Once it moves the actin and is in this conformation here, then it's able to bind ATP. Then hopefully it's there, right? Because muscles are very well supplied with blood, right? Muscles, you cut the muscle, it's bloody, right? So ATP is there. Once ATP binds, it's going to cause dissociation of the head to the myosin. That is a cool thing, right? So now this part here of the head of the myosin has an enzyme that is ATP hydrolysis. It can break down ATP. Okay, so now to the next stage here. This part here is going to have ATP hydrolysis. Make sure you watch the little video that is in your homework, right? ATP hydrolysis breaks down the ATP. When ATP breaks down to ADP, so now the head goes back to the original conformation, and now it can bind to actin again. Okay? So it binds to actin again, it's going to make the bridge again. All right? So here, important thing to know, the ATP hydrolysis is going to fuck the myosin head. Sorry, that's the word they use. What other word to do with it? It's down, it goes up. You never forget that, right? All right, ATP hydrolysis is going to do what? It's going to get the head of the myosin up. It takes energy. It makes sense. ATP hydrolysis, right? Okay. So once you get this thing back up, it's going to bind to actin. And then again, binding to actin, you're going to have your kicking out ADP. You're going to bind ATP again. That is going to, kicking out ADP is going to make the thing to turn, push again on your actin. Then you bind ATP again, the thing comes down again. Okay, did everybody get that? Oh, look at that. More questions. Oh, you got a whole inch, flow the questions today. What is calcium's function during muscle contraction? And your answer is A. Remember, calcium is going to bind to troponin. Remember, troponin doesn't have myosin on the name, right? Calcium is going to bind to troponin. It's going to change the shape. It's going to remove the block of troponin, of tropomyosin that is on actin. All right. So here, just to put in one page what's going on with this cross bridge formation, so you have a little summary there. Make sure you understand that. Here is still another demonstration, another view of how this system is working. Okay, so make sure you understand that. Okay, so here's a little movie that shows in a beautiful setting of 3D thing how this thing is working. You can watch that on your own. So here, when you're going to trigger a contraction, so calcium is really the key for the contraction, right? How is it that calcium is going to get to the sarcoplasmic reticulum? You have active transport pumps that are going to work in there, okay? Now, this is the part that I like about this, all right? Drugs that act on your neuromuscular junction. My favorite, botulinum toxin. Who heard of this guy before? Yeah, where did it come from? I mean, the thing. Yeah, where is it coming from? Bacteria is right there. It is a bacterium, botulinum toxin, right? Very powerful bacteria, okay? So what is it gonna do? Okay, we heard of Botox. What is Botox used for? Make you younger? No, it kind of prevents aging. But how? Well, like some people like to inject some muscles in the forehead, right? Because muscles in the face, like they attach to bone, not to bones, to skin, right? So here, if you use a lot the muscles in your forehead, after a certain time, you're gonna get wrinkles, right? So some people decided that putting some botulinum toxin on their forehead is going to avoid the muscle from contracting and is going to avoid wrinkles. Right? I'm not sure if that's a good idea. Yeah.
I heard somewhere it's also migraines as well.
Yes, migraines, because migraine sometimes is a muscular tension that causes migraine. But for the migraine, you get injections in the back here of your head, not in the forehead. Yeah, it's like here somewhere. I know I got migraines and those are not fun, believe me. Definitely not fun. Okay, so how is the mechanism? How is this thing is working? Okay, Botox, botulinum toxin, blocks exocytosis. What is exocytosis? Vesicles. Vesicles fusing to the membrane. Which vesicles? The ones that are carrying acetylcholine, right? So when this thing is working at the neuromuscular junction at the axon, the nerve. So you block these vesicles from fusing to the membrane. You do not dump acetylcholine in your neuromuscular junction at the synapse, and that prevents muscle from contracting, okay? So it affects the nerve. Now, it affects the nerve at the neuromuscular junction. Now, botulinum toxin is very powerful, very, very powerful. If you put more of it than you should, it should be, by the way, very, very diluted. If you put more of it than you should, it can affect nerves that control some very important muscles like your diaphragm, breathing muscle. And what does that mean? Yeah, that means your diaphragm is not going to work. You're going to stop breathing and you're going to die. Were there deaths associated with Botox usage? Yes. Was there paralysis associated with Botox usage? Yeah. So I don't know if I like this idea that much. Okay. So what is the mechanism? Blocking acetylcholine release, blocking the exocytosis. All right. So that's Botox. You got to know Botox. Okay. Everybody knows Botox. All right. So here, next one that not a lot of people are aware of, nicotine. What does nicotine do? There we go. You can just read this line. It's going to stimulate the ligand-gated receptor. If you know anybody who smokes, okay, I'll tell you from not personal experience, but secondhand smoking. My mother smoked her whole life. I think half of my life I tried to convince her nicely to stop smoking and it never worked. Oh, I can tell you stories. Oh my God. Anyway, so what happened? Smokers like first thing in the morning to have a little coffee and to have their cigarette, and then what happens? You don't know anybody who smokes? They have coffee and a little cigarette, and then huh? They go to the bathroom. It's a ritual with the smokers. Little coffee, little cigarette, smoking, and then the bathroom time. It's not a coincidence because nicotine is going to stimulate cholinergic receptors and then makes all these muscles work a little better and a little faster, right? No. Smoking will kill you faster than you should, okay? Don't smoke anything. Anything you're ready to smoke is not good, okay? So nicotine, that's an interesting one, activates, stimulates your cholinergic receptors. Now there's another one here, curare. Okay? This is a poison that blocks the receptors. Curare causes flaccid paralysis because if you block the receptor, it's almost the same thing as blocking release of acetylcholine, right? So if you have an overdose of Botox, you're also going to have flaccid paralysis, okay? So this poison and that poison have the same effect but different mechanisms, okay? This one blocks the receptor, this one blocks exocytosis. Both of them cause flaccid paralysis. Muscles do not contract. On the other hand, you have other poisons like sarin and organophosphates that they cause blockage of the enzyme that breaks down acetylcholine. So now you're going to have a lot of acetylcholine at the synapse and constant stimulation of the muscle. What's going to happen to the muscle? Spasm, muscle spasm. So that will kill you by spasmic paralysis. Do you know where or when sarin was used as a biological weapon? I think it was World War I. Nerve gas, yeah, the sarin. And I think it was used not too long ago in Syria. I'm not sure, I forgot. Maybe Syria, right? Yeah, yeah. Although it is illegal, but a lot of things are illegal. All right, so sarin, nerve gas, right? And also this other thing, what is an organophosphate? Pesticide. Who uses organophosphate? Everyone who has a farm and is not using organic type of pesticides. Scary. If you work in a farm and you're exposed to high levels of organophosphate, guess what's going to happen? A lot of stuff is going to happen, including spasmic paralysis. Okay, not good for you. Okay, so problems that are associated with this neuromuscular junction. One of them is myasthenia gravis. Okay, this is a disease in which the receptors do not work very well. So the receptors are damaged, and then the muscle doesn't really work very well. So you get weak muscles. That's myasthenia gravis. Now, a lot of people have heard of rigor mortis. This is when you have a cadaver that is dead for about 12 hours or so, right? and you try to move the thing and it's like stiff. That's rigor mortis. What happens when the person gets stiff after death, because you don't have any more ATP that is going to release the binding of the cross bridge. Cross bridge binds myosin and actin and keeps it there, keeps the muscle contracted, okay? Now, ATP, if your blood is flowing and you have nutrition and all this oxygenation going on, ATP is going to break the cross bridge, right? Since there's no more circulation, the person is dead, the thing is dead. So you have all these fibers, muscle fibers locked in place. So that's what causes rigor mortis. So we're going to stop here.